It hunts with light. Its prey cannot see it, not because the light is dim, because the prey's eyes are physically incapable of detecting that wavelength. Malacosteus niger, the black dragonfish, 20 centimeters long, teeth like curved glass, skin so dark it registers as an absence rather than a color. It lives between 200 and 1,000 meters down, in a zone where sunlight never arrives, and pressure would crush an unprotected human lung in seconds. In 1983, researchers measuring the bioluminescent output of deep-sea fish recorded something strange. The dragonfish's light organ, a small structure positioned just beneath each eye, was emitting at a wavelength far beyond what any deep-sea animal was supposed to produce. The numbers didn't fit the model. The data was logged. The significance wasn't understood yet. It would take 15 more years for someone to ask the right question. The right question wasn't, "What is this light?" The right question was, "Who can actually see it?" In May 1998, Ronald H. Douglas at City University London and Julian C. Partridge at the University of Bristol published their answer in Nature. What they found stopped the field cold. The dragonfish wasn't just producing unusual light. It was producing light that, as Partridge and Douglas wrote directly, apparently only it can see. A private frequency, in the deep ocean, running continuously. Edith A. Widder, documenting deep-sea predator-prey dynamics in 2005, confirmed the mechanism's ecological consequence. Most deep-sea fish are, in her words, essentially blind to wavelengths longer than 650 nanometers. The dragonfish hunts beyond that threshold entirely. The prey fish hover in the dark, lateral lines active, eyes forward. They detect nothing. No pressure wave, no photon, nothing. But buried inside the dragonfish's own retina is a molecule that should not exist in any animal's eye, a molecule that explains everything. The exact identity of that molecule, and the single peer-reviewed discovery that cracked it open, is where this goes next. Here is the simple explanation. Red light disappears in seawater faster than any other wavelength. By 2 meters downward, it is gone. So when researchers first measured the dragonfish's bioluminescent output near 705 nanometers, the obvious conclusion was camouflage. Passive. Accidental. Almost. The logic was clean. Every other predator in the mesopelagic zone produces blue-green light, around 470 to 490 nanometers, the only wavelength that survives depth. Red light dies before it reaches anyone. So why would prey evolve sensitivity to a frequency that never reaches them? They didn't. Case closed. Except the case wasn't closed. In May 1998, Ronald H. Douglas at City University London and Julian Partridge at the University of Bristol published a paper in Nature. They weren't just measuring the dragonfish's light, they were measuring its eyes. What they found should not have been possible. The dragonfish retina contained photosensitizers, pigment molecules tuned specifically to wavelengths above 700 nanometers, not blue-green, far red. The same frequency its own suborbital photophores produced. Douglas described it precisely. The dragonfish has effectively created a private waveband for communication and prey detection. This wasn't camouflage. The animal could see its own light. Nothing else could. ROV footage confirmed what the lab data implied. Within one body length of prey, the suborbital beam was active and directed forward. The prey showed no startle response, no evasion, no awareness at all. The dragonfish was illuminating its target in a frequency the target's nervous system simply could not register. Partridge stated it plainly. This was, he wrote, the only known case of a bioluminescent organism producing light that it, and apparently only it, can see. The passive camouflage theory was dead. The dragonfish wasn't hiding its light, it was using it deliberately, at close range, as a targeting system. But that answer opened a harder question, the one no one could answer, in 1998. Where did the retinal pigment come from? Here is where the story breaks open. The dragonfish did not evolve a new photoreceptor from scratch. It stole one, not from another animal, from a bacterium. In 2000, researchers examining preserved Malacosteus niger specimens found something that had no business being inside an animal's eye: bacteriochlorophyll, the same photosensitizing molecule that photosynthetic bacteria use to harvest sunlight in a fish. In a retina, at 800 meters below the surface, where no sunlight has ever reached. The pathway is a chain. Photosynthetic bacteria drift in the water column. Copepods, crustaceans barely 2 millimeters long, consume them. The dragonfish consumes the copepods. Somewhere in that digestion, the bacteriochlorophyll survives. It migrates into the retinal tissue, and it transforms. Inside the dragonfish eye, the molecule is repurposed. It becomes a photosensitizer, a converter that shifts the retina's sensitivity from the standard deep-sea range, around 470 to 490 nanometers, up past 650, past 680, all the way to 705 nanometers, far red. The exact wavelength the dragonfish produces from its own suborbital photophore. A spectrophotometer run on dragonfish retinal tissue, documented in the 2000 follow-up to Douglas and Partridge's 1998 Nature paper, showed a sharp absorption peak at 670 nanometers. That peak is the bacteriochlorophyll signature. It is not supposed to be there. It is the only recorded case of a dietary molecule being incorporated directly into a vertebrate visual system and functionally rewiring it. Partridge stated it plainly. This is the only known case of a bioluminescent organism producing light that it, and apparently only it, can see. By 2014, Sonka Johnson at Duke University had surveyed mesopelagic fauna comprehensively. No other deep-sea predator shared this capability, not one. The dragonfish did not invent a new frequency. It found a discarded key, bacterial, ancient, photosynthetic, and cut a lock around it. Every animal around it swims in what it believes is total darkness. The dragonfish moves through a lit corridor only it can perceive.